Hi everyone, I'm Connie Wu, a postdoc in Dr. David Walt's lab at Brigham and Women's Hospital and the Wyss Institute for Biologically Inspired Engineering. Today I'll be giving a general overview on COVID-19 diagnostic testing and the current challenges they face, and I hope this will give you some good ideas for creating your games. COVID-19 began spreading quickly in the US in March, and since then, we have not been able to get control of the number of cases and are still recording about 40,000 cases a day as of the beginning of September. As society reopens, the most important problem will be, how do we keep the number of cases from increasing? One of the most important keys to flattening the curve of COVID cases and keeping it flattened as society reopens is diagnostic testing, which has been one of the most critical challenges in our fight against the pandemic. To understand how current COVID-19 tests work, let's first look at the virus. Each viral particle consists of proteins and RNA. Several of these proteins make up the virus's structure, including spike proteins that cover the outside surface of the viral particle. The virus's RNA, its genetic material, is protected inside the viral particle. These protein and RNA components can provide different targets for detection in COVID-19 tests. Current COVID-19 tests can be broadly categorized into three types based on what they're measuring. The most commonly used test measures parts of the viral RNA. A second type of test measures the levels of antibodies that a person's body has produced against COVID-19. And finally, there are a growing number of antigen tests which measure the virus's proteins. Currently, viral RNA tests remain the standard tests for diagnosis of COVID-19. Most RNA tests use a swab to obtain a sample from the back of the nose and throat, or from the front of the nose, which is then transported to a laboratory, where the RNA is extracted and measured. So what has prevented testing from effectively containing the pandemic? There are several major challenges, including sensitivity, testing speed, and availability of testing. As an example, to demonstrate why the sensitivity of a test is important for containing viral spread, let's look at a group of 10 people who have been infected with COVID-19. Current RNA tests for COVID have been estimated in some cases to be 70% sensitive. If we have a test with 70% sensitivity, this means that 7 out of 10 infected people on average will test positive. Upon testing positive, these people will be quarantined, which will prevent further spreading of the virus. But a 70% sensitive test means that 3 out of 10 infected people on average will test negative. These people will therefore not be quarantined, and if they are still in the stage where they are actively infectious, these undetected individuals can potentially spread the virus, which can quickly grow into a cluster of cases if not effectively contained. Another challenge is the long testing turnaround times. Often the samples must be transported to a central laboratory for testing due to the complex workflow and equipment needed. For RNA testing, the test itself takes several hours. Altogether, it typically takes at least 1 to 2 days, and often longer, from the time of sampling to getting back the results. The need for expensive laboratory equipment and trained personnel also make it difficult to ramp up testing. As society reopens and there are more opportunities for exposure and viral spread, we will need much more frequent and widespread testing, which has been a major challenge. To address these challenges, there have been many efforts to develop point-of-care diagnostic tests for COVID-19, either measuring viral RNA, antibodies, or viral proteins. These tests are portable and can be performed at or near the site of sampling, which can improve testing speed and throughput. Hopefully, I've convinced you of the importance of diagnostic testing in fighting this pandemic. To help society reopen safely and crush the curve for COVID cases, we need tests that are accurate, fast, and widely available. Currently, there are many research efforts across the world focusing on improving all these aspects of COVID testing for measuring all 3 types of molecules, either viral RNA, antibodies, or viral proteins. For more information on the latest COVID-19 tests that are being developed or newly approved by the FDA, please see the following links. Thank you for your attention and good luck on the game jam.